These are notes on Unit 5, which is about the cell membrane, transport, and energy, covered in Chapter 5 of your textbook. Part 1 is about the cell membrane and passive transport. There's a vocabulary words that will be used throughout the unit, so make note of these before we get started so that you'll know which words you're responsible for. The vocabulary words are in red on the slides, and most of the things that you need to write down are in purple. So the plasma membrane, as we talked about in our last unit, which was the structure and function of the cell organelles, forms the boundary of the cell and controls what enters and leaves the cell. That's its main function. The way it does this is because it's called what we call selectively permeable. That means it lets some molecules pass, but not others. The thing you need to realize about the membrane is it's not just a phospholipid bilayer, as we talked about before. We call this the fluid mosaic model. It also contains some other molecules. It contains cholesterol and various kinds of proteins that are embedded in the membrane. The cholesterol helps keep the lipids from getting too solidified or too tightly packed when it's cold or too loosely packed when it's warm. And the proteins have various functions, and we'll talk about the functions of some of those proteins as we go through the unit. Please realize that the plasma membrane is the same kind of membrane that you have enclosing the organelles in cells. So when you have a vacuole or a vesicle of some kind that has a membrane around it, it's the same type of membrane. When you have endoplasmic reticulum or Golgi apparatus, they're made of the same kind of membranes that you find on the outside of the cell. The phospholipids are the main component of the phospholipid bilayer, the cell membrane. Realize that the phospholipid is a lipid. It's made of fatty acids. Here's some fatty acids and glycerol. But instead of 3 fatty acids, it has only 2. And attached to that third carbon in the glycerol molecule, we have a phosphate and another molecule. This part of the molecule then becomes polar. And polar molecules, as we've already learned before, are what we call hydrophilic. They like water. Remember that fatty acids, being hydrocarbons, do not like water. They're what we call hydrophobic. They're nonpolar. And so that makes what we call an amphipathic molecule. It has a polar end and a nonpolar end, similar to what we've seen in some other molecules we've talked about before. Usually at least one of these hydrocarbons chains has a double bond, a carbon-to-carbon double bond, which causes this little kink. And that's important because it helps keep the phospholipids from packing too tightly together, because we need to be able to get things through the membrane that we need to get through there. So draw the model, this model over here, number C on the right hand side in your notes, just to show that and label it so that you can see the polar hydrophilic head and the nonpolar hydrophobic tails. Now, if you put a lot of phospholipids into a beaker of water, you're going to have an automatic formation of this phospholipid bilayer. Because these hydrophobic tails want to do everything they can to stay away from water. And so they'll arrange themselves with the tails together and the hydrophilic heads on the outside so as to keep the water out of this region. Because the water, remember, is very polar, so it doesn't like to mix with this nonpolar part. This is really nice because it allows us to have a separation between 2 different solutions. So draw a little bit of the membrane, a couple of phospholipids, a couple of the phospholipid pairs, the bilayer here, and label the hydrophilic polar part and the hydrophobic nonpolar part of the membrane. Now the membrane has a lot of proteins embedded in it, as we said. And some of those proteins do different kinds of jobs. You can see in this section of membrane here, you can see that it's made of a bunch of smaller molecules. That makes it a mosaic, and it's fluid because it's flexible. These various proteins are embedded in and attached to the membrane. Some of them are surface proteins on the upper or lower surface. A lot of them are transmembrane proteins, meaning they go through the whole membrane. So here are some of the kinds. We have channel proteins that can transport certain molecules across the membrane from one side to the other. We have some receptor proteins that receive signal molecules from other sources, and we have other proteins called glycoproteins that have carbohydrate chains attached to them that act kind of like identification tags for the cell. Now, the the main reason that you need a membrane, and the main job that the membrane does, is to help the cell maintain homeostasis. Remember that all living things attempt to maintain a stable internal environment, a steady state. They are regulating this environment in a lot of different ways, and a lot of the functions of the membrane have to do with maintaining homeostasis. It is very important because it allows, certain molecules to come in, certain molecules to go out, to keep things the way they're supposed to be. There are lots of different activities that organisms do to maintain homeostasis, and oftentimes you can say just about any activity the organism carries on is directed toward maintaining that stable internal environment. So how do cells maintain homeostasis? Well, there are a couple of different ways they do this. There are several different processes. There's passive transport and active transport, the two major categories. Passive transport is the movement of molecules that will happen on their own. There are three main kinds of passive transport: diffusion, osmosis, and facilitated diffusion, and we'll talk about each of these in turn. The other type of transport is called active transport, and this is the type of transport that requires energy. It requires some work to be done. There are proteins that actually use energy to move molecules from an area of lower concentration to an area of higher concentration. There is a process called endocytosis, moving things into the cell, and another called exocytosis, moving things out of the cell. And we'll talk about active transport in the next section of the notes. Back to passive transport, though, the first kind we'll talk about is diffusion. By definition, diffusion is the movement of molecules from an area of high concentration to an area of lower concentration. Molecules never stay still. They're always going to be moving around from place to place. They bounce off each other all the time. When you have a closed system, as we see here in this box, okay, and we have a high concentration of solute molecules at one end, because the molecules that are in here, the water molecules and so forth, are moving and bouncing around, they're going to start knocking into these, and that's going to cause these to move. Eventually, they're going to spread out to fill the whole space more or less evenly. But when you start from an area of high concentration, generally speaking, the molecules are going to have a tendency to move from high to lower concentration, and we call that the concentration gradient. You'll hear the term moving down the concentration gradient, along the concentration gradient, with the concentration gradient. They all mean the same thing, moving from high concentration to lower concentration. So here we see what happens if you put a membrane that allows some molecules to pass through in a beaker of water. You put dye molecules on one side of it, and they're moving around and bouncing around. Some of them are moving to the left, some are moving to the right, but generally speaking, more molecules would be moving over to the right, passing through the pores, than moving to the left. Same thing here, we have more diffusion going back over toward the right, although there are some molecules moving back to the left because they're always in motion. Finally, after a while, you'll reach an equilibrium point where you have more or less equal distribution of the molecules on both sides of the membrane. Now, another type of passive transport is something called osmosis. Osmosis is a type of diffusion as well. It is still movement of molecules from a high concentration to a lower, but it specifically is the diffusion of water down its own concentration gradient from high concentration of water molecules to lower concentration of water molecules across a selectively permeable membrane. So here we see the membrane in the middle of our system, and we've got a high concentration of water molecules on one side, a lower concentration on the other side, and the tendency will be for more molecules to move to the right across the membrane until you reach the equilibrium point where they're moving equally back and forth. Now, one thing you need to understand about the water movement as it occurs in cells is that, again, we're talking about movement of water from a high concentration of water molecules, but what we're really talking about is a high concentration of free water molecules. When you have substances dissolved in water, some of the molecules of the solute are what we call bound to water molecules, or the water molecules are bound to the solute molecules. They have to stay with them. And other water molecules are not bound to the solute molecules. They're what we call free water. And so actually what's happening here is that you have a higher concentration of free water, and those are the molecules that are going to move across the membrane. And so keep that in mind as we talk about the types of solutions. When you talk about water balance, you're talking again about the movement of free water from one side of the molecule to the other, from the membrane, across the membrane. So there are three main types of solutions based on the concentration of solutes. that affect the movement of water molecules in the cell and out of the cell. The first type is hypotonic. A hypotonic solution is one that has a lower concentration of solutes than the cell has. That means the cell has less free water and there's more free water outside the cell. And so what's going to happen there is that water molecules, the free water molecules, are going to move into the cell. The cell will take on water, and that's going to cause swelling of the cell. Again, you have a lower concentration of solutes in the solution than you do inside the cell. And hypo means under, remember, so you've got a less concentration of solutes in the solution than you have inside the cell. Isotonic solutions, iso means the same or equal, just like an isosceles triangle has two equal sides. This is a solution that causes no net movement of water from one side to the other, either into or out of the cell, because the concentrations of solute particles are about the same inside the cell as outside the cell. The cell is going to remain the same size, and there's not going to be any net movement. Will there be water movement into and out of the cell? Yes, there will, but it'll move more or less equally from one side to the other. The third kind of solution is a hypertonic solution. Hyper is too much or above, and this is a solution that has a higher concentration of solutes than the cell has, and that means there's more free water in the cell than outside the cell, and so this is going to cause the cell to lose water. And this is going to, when the cell starts losing water, it's like you had a water balloon that you poked a hole in with a pin. It's going to start getting smaller. It's going to start shrinking up. Here we have two different kinds of cells in these three different kinds of solution. Let's talk about the animal cells first. If you put an animal cell, like a red blood cell, into a hypotonic solution, then more free water is going to be outside the cell than in, and so water is going to move into the cell, causing it to swell, and eventually it will rupture or lyse. When that occurs, the cell will die. In an isotonic solution, water is going to move into and out of the cell equally, and so it's going to be in a normal situation, and the cell is going to have its normal shape and size. If you put that cell in a hypertonic solution, there are more dissolved solutes outside the cell than in, which means the free water is going to move from inside the cell to outside, and that's going to cause the cell to shrivel or shrink. Now in a plant cell, it's a little bit different because plants really like to be in this hypotonic state. When there's plenty of water with a plant, it's going to stand up straight and tall. Okay, there's going to be extra water inside here, which is going to exert pressure on the interior of the cell walls. This is called turgor pressure, and that makes the cells turgid, which is the normal condition for plant cells. They like to be like that, so when you see a plant standing up nice and straight and tall, because it has plenty of water, it's in that hypotonic state. When the plant starts losing water, let's say you forget to water the house plants, or we're in a drought situation, what happens to the plant is it starts to lose water from the inside, and that reduces that pressure on the inside, and it causes the plant to start wilting. Because even though it should be normal, as we would think here in the case of the animal cell, for plant cells, you're not quite so happy being in that state. And then if you get the plant into a hypertonic solution, what is going to happen is more water is going to leave than is going to remain in the cell, and you'll end up with the cell becoming plasmolyzed, meaning that it's going to shrivel up, and you're going to see the plasma membrane pull away from the cell wall. That's about the only time you see the plasma membrane in a plant cell, because the rest of the time it's up against the cell wall. So when you look at these cells, here's a cell. The green dots here are solute molecules, and this is the solution on the outside of the cell. So this is a hypotonic solution, so that means that water is going to move into the cell, causing the cell to swell. This is a hypertonic solution because there's more solute outside the cell and more water inside the cell, so water will move out, causing the cell to shrink. And here we have an isotonic solution where the water is going to move equally into and out of the cell because the concentrations are equal on both sides of the membrane. Now, another type of diffusion is called facilitated diffusion. This is also passive transport. Facilitated means made easier. And what you need to realize is that there are some molecules that are either too big or have too strong of a charge, either positive or negative, to pass straight through the membrane. The size you can understand. If they're big enough, they're not going to fit through. The polarity is something else, because remember, if they have a positive charge or a negative charge, that makes them polar, and that means they're not going to be very happy or the phospholipid tails are not going to be very happy coming into contact with them. So they have a tendency to block them out. Those molecules need a special kind of transport protein called the channel protein. The channel proteins have an opening in the middle that allows certain solutes to pass through. They're still moving from a higher concentration to lower, so it's still diffusion, but they're facilitated or made easier to go through this protein that allows them to pass through. There are simple channel proteins like this one shown here. There are some that are called gated channels that have a special molecule that fits in a space that causes them to open up for the molecule that they can pass through. But none of those requires energy, so they're still diffusion. They're still moving from higher concentration to lower. And that concludes our discussion of the membrane and passive transport.